Thank you. Good afternoon. My name is Mahmoud Parsian, and I will be talking about allelic frequencies in genomic data. This is a very specific application in the genomic business using Hadoop. And some of the information that we got yesterday on some of the presentations, they were not very correct, so I want to correct some of those data. They were talking about that DNA sequencing takes about 700 to 800 megabytes of data. That's not true. I have the actual data that I'm going to read from here. A patient sample, such as a saliva or blood sample, if you give it to the shotgun sequencing and generate the DNA sequence data, that will be about 250 up to 750 gigabyte of data. It is not 700 megabyte or 800 megabyte. That's the first correction I want to make. And then once you have that raw data, 750 gigabyte up to 750 gigabyte, you go through DNA sequencing analysis, which you do alignment, recalibration, and finally finding the variance. That will generate another 1 to 2 terabyte of intermediate data. Eventually, the DNA sequencing will generate between 200 to 400 megabyte of variant data, which then the data is ready to be analyzed. So what's the variant-based data, which we are going to talk about here, is that the interesting part. All of the human genomes, we share the commonality between the human beings. 95% of the DNA are the same. Only 5% is different among all of the humans in the world. Only 5%. That's what we are going to focus on, the allelic frequencies, to see that even with that 5%, what we can do. And what pharmaceutical companies are doing to using these Hadoop technology to basically find out what are those allelic frequencies and how they can help the patients. This is basically the agenda for my talk. Will be first I'm going to talk about a minute about myself, what am I doing and what do I do, and next I will give you the overview of the allelic frequencies and what does it mean, and in simple terms. I'm going to try to stick with simple terms so that we can all understand. I know that you guys are not biology experts. Probably most of you are the computer science experts, as I am also. I'm just learning biology myself. So I'm not also a biology expert. So when I have a question, I go to our bioinformatics team and validate the notions that I have learned, so on. I'm going to go through some of the basic definitions of the allelic frequencies, and then I will talk about what is the source of data, what kind of input we are going to be using for this analysis that we will do. And then I will talk about what is the meaning of the allelic frequencies using the Mapreduce Hadoop. I will show you that later on. I use a 3-phase Mapreduce algorithm to solve this problem, basically. And then finally, I will show a sample run that how this allelic frequency is run. As I said, my name is Mahmoud Parsian. I have a PhD in computer science. I have been using Hadoop since its inception. I started Hadoop with a 3-node cluster of weird nodes, and now finally in the production we are running with several hundred nodes, basically, at Illumina. I lead the big data team at Illumina, and we are using our goal for the company is that to develop scalable algorithms using the DNA sequencing, RNA sequencing, and regression algorithms to basically help the canceric patients to solve some of their problems. Next, I'm going to talk about the overview, what we want to do. As you see from this slide, is that the patient sample, from a patient sample, we create a VCF. What's a VCF? It's a variant call format. It is those are the differences, the 5% that I was talking about, that's expressed in the variant call format. So the patient sample, such as a blood or saliva, will go through the shotgun sequencing. It generates the DNA sequencing up to 750 gigabyte of data. And then after that, we will go through the alignment, recalibration, SNP detection. Finally, we will create that VCF. Just one example, if you have a decent server, DNA sequencing for one single sample might take up to 72 hours. Using Hadoop, we bring generation of that VCF to under 1 hour. So that is the power of Hadoop right there, to just generate the VCF file for a patient sample. And the magnitude of data is so huge. For one single patient, we have about 4.3 million records in the VCF. 4.3 million. And if you do the ingestion on that VCF, you will generate up to 20 million records for that just single patient. So the magnitude of data, if you want to analyze 10,000 patients, compare that to maybe 3,000 normal human samples, that will generate magnitude of billions of records. So that is how Hadoop is helping us to solve this problem. So the magnitude of data is huge. Sometimes in pharmaceutical companies such as Sanofi or Merck, they compare about maybe 100,000 patients with another 50,000 basically healthy samples. And then to do that regression algorithm, it will come up maybe about 100 to 200 billion records. So the Hadoop in this case is helping a lot in that case. For the allelic frequency, whenever we hear about the frequency, we may be thinking about the word count frequency, right? That's a little bit different from that. Yes, we are finding the frequency of the alleles, but we do not just count one by one. We generate our different tables and then use different regression algorithms. In this case, we are going to use a Fisher test, Fisher's exact test, which is a binomial algorithm, and I will show you that what is that algorithm. It's a very hard computation, so that you have to do several, basically the factorial algorithms to solve that Fisher exact test. So, as I talked, the magnitude of data is very challenging to analyze. We have several million variants per patient and several billions for groups of patients. And then the Mapreduce, we find the allelic frequencies. And finally, we say, what are the top 100 which are closer to zero? Zero means that those frequencies are very important to a physician or lab technician that they are interested in. And again, for all of those, we are using the Mapreduce Hadoop. Before going into the allelic frequency and genomic data, let's talk about just a little bit biology information to see what those terms mean and so on. So the basic definition of chromosome, Bioset, Bioset record, allele, and allelic frequency. So these are the basic definitions I would like to go. And I'm sure most of you have had biology classes. You know what is a chromosome, but maybe some of you don't know Bioset and Bioset record. As you know, the chromosome comes from a Greek word, and basically it means the Greek words for the color and also the body. And human human body is composed of 23 chromosomes, and they come as a pair. One from father and one from mother. Basically, we get one from each parent. So that becomes basically a couple of 46. So that's a chromosome. And this is basically the chromosomes in picture. We have 23, and X and Y are the sex chromosomes. And this is the cells to DNA. Basically, we have trillions of cells in our bodies, and each of those cells basically translates into the DNA. And then the genes are composed of basically hereditary genes are basically in the DNA. And the DNA code, as you know, it's the basically code of a human being. It is composed of the A, T, C, G, and I'm not going to go the abbreviation for those, basically those are the DNA code, A, T, C, G. And they come with a pair, A with T and C with G. What is a bioset? When we talk about the analysis, bioset means that, okay, we have generated the variant call format, and now we are ready to analyze that data. But before analysis, we need to understand that how we are going to assign the mutation classes for each record. How we are going to assign the genes from the database. How we are going to compare those to the references, to the human genome references. So that's called the ingestion process. You get a VCF, you go through the ingestion process, assign mutation classes, assign gene IDs, and assign different attributes based on whatever you know so far. That is called the ingestion process. So the bioset is basically, we ingest and we find out what are those mutation classes, what are those genes, and that's called the bioset, basic. Still, it is not analyzed, it is just a very fundamental data. Now, after bioset is created, we can do different analytic algorithms, such as regression testing, or such as allelic frequencies, and so on. So bioset is just a VCF in a normalized fashion. What does a bioset record look like? It has a chromosome, it has a start position, stop position, and 2 alleles. And where those alleles are coming? One from the father and one from the mother. Allele 1 and allele 2. For example, the example I have listed here is for the chromosome X. For the sex chromosome, it has a start position, end position, and G is the allele 1 and A is the allele 2. So the basic of the bioset is that it has 4.3 million records, and once you ingest that, that will become maybe up to 20 million records, because each of those rows, once they are ingested, it can create multiple entries for the mutation, different mutation classes. This slide is showing basically that how we are creating. So a patient sample generates a FASTQ format. It's a text-based format which has the DNA sequencing and the quality of data. And then that FASTQ file, which is the size of that can be about 250 to 750 gigabyte, goes to the DNA sequencing and generates a VCF file. And DNA sequencing by itself is a big workflow, as I mentioned, alignment, recalibration, and then the variant detection. And then finally, we convert VCF into Bioset, and then Bioset is converted to a germline ingestion. We ingest that to find out what are the mutation classes, what are the genes, and so on. And then once you have the Bioset, now you are ready for the different algorithms, analysis algorithms, regression algorithms, allelic frequency, or other algorithms that you are desired. Or there is another test which I'm right now working on, is called a Cochrane Armitage test, basically they are using for detecting the cancer in the pharmaceutical companies. So that's another test that can be used. Just summarizing the Bioset record. So Bioset record has chromosome, starting position, stop position, 2 alleles, genomic reference, and other related information such as the mutation class and so on. And what is an allele? So allele is a viable DNA coding that occupies a given locus on a chromosome. And there are 2 alleles, as I mentioned. One comes from a mother and another one from father. So 2 alleles. And this is the picture of the basically chromosome with 2 alleles. What is allelic frequency? The allelic frequency is defined as the percentage of the population of the species that carries a particular allele for a chromosome locus. This is important for the physicians and the lab technicians, as well as for the pharmaceutical companies that find out how frequent is that allele for a given chromosome in a sample of patients. And then finally, once you find that frequency, you apply an algorithm, Fisher's exact test, to find what is the allelic frequency. The next, I will talk about the source of data for allelic frequency, that in order to do the allelic frequency, what kind of data we need. As I mentioned before, so we have a sample of patient sample, converts to FASTQ file. FASTQ file converts to DNA sequencing, and finally to VCF, finally to Bioset, and then once you have all these information, so these 7 information are the foundation for most of the regression algorithms and other algorithms that you can use. You have to have those information in order to do any viable analysis. Without those, the result and analysis will not be meaningful. You need to have those 2 alleles, a starting and ending position, and the reference genome and the mutation class. Let me give you the next slide is talking about the size of the data. What is involved in this analysis? I have given a very simple example here showing that one bioset is 4.3 million records. And to do the allelic frequency, you have group of A and group of B. And note that in group A and group B, I can use the same biosets. I can select. I might. So in order to represent this data in the Hadoop, we generate the same data using group A and the same data with group B. So almost we have a duplicate data, but the only difference is the one field, A or B. And the reason we did that for the efficiency purposes, of course we could have generated just one set of data and at the runtime to assign A and B, and that will take more time. But we generate 2 sets of data identical, but the only difference is in the single column called A and B. Because different people can use these data for different kind of analysis. And here I have given a very typical example, group A considering 6,000 biosets, group B having 9,000 biosets. So you have 15,000 biosets, and that translates to 64.5 billion records. In real tests for pharmaceutical companies, the minimum number of samples they might select, it will definitely bypass 20,000, 30,000. This is a very small sample I'm mentioning here. Next, I will talk about the allelic frequency analysis. What does that mean? Okay, what's the problem and what we are trying to find? Given 2 groups, group A is a set of biosets, and group B, another set of biosets. we want to find allelic frequency for every chromosome ID, start and stop an allele. So that will be your key. So the key will be the chromosome ID, start, stop an allele. And then the goal is to find the p-value, the probability from 0 to 1, how close that is to the 0. The more closer it gets to 0, it is more interesting to the physicians and lab technicians. And finally, we will find the top 100 p-values. Top 100 p-value means the actually bottom 100 because we are finding closer to the 0. So this is basically the allelic frequency analysis in a nutshell. Given a group of biosets, another group of biosets, we want to find what is the frequency of specific allele for a specific chromosome at the start and stop position. I'm going to go with an example so that we can comprehend what does this mean. I'm going to go with let's say our bioset, we have 6 biosets in group A, and bioset IDs are numbered from 1 to 6, and allele 1 and allele 2, so this is very simple example for the group A biosets. The group B, suppose it has only 5 biosets, and they are identified from 7 to 11, and again we have given allele 1 and allele 2. Next, I'm showing you that what is the allelic frequency means. We have an allele, on the leftmost column, we have allele A, C, G, A, C, T, and then if you look at the orange columns, the total has to be 12, right? 6 times 2, because I had 6 biosets. And if you look at the last 2 columns, the total has to be 10. I had 5 biosets. So it says how many of those, for example, allele A, how many of those were present in the group A, and how many were not present in group A? Yes or no, basically. Okay? So if you look at the allele A in the group A, 6 of them were there, and 6 of them were not there. And the same as for the others also. So the total of those has to be double the size of the number of biosets. Make sense? If you take just one single allele, A, and basically it has known positions and others, and the group B, 7 and 3. So 6 and 6 becomes 12, double the size of the first bioset group, and 7 and 3, 10, 2 times 5 is 10, basically the total, the double size of the groups in the bioset B, group B. This is called a contingency table. Once you have this contingency table, then you can apply the Fisher's exact test. And then that will give you the p-value for every. So going through all of the data, the goal is to form this contingency table and finally apply the Fisher exact test. And if you know the R programming language, you can use, by the way, R is the golden reference for the bioinformatics people. And even some of these, I could not find any Java package to do the Fisher's exact test and so on. So sometimes even in the Hadoop, we call R to just do the calculations, because that's the golden standard for the bioinformatics. And here, it is the sample I'm showing that how we are doing the Fisher's exact test using the R. I format 2 tables, 6 and 6, the counting the frequencies, and 7 and 3 for allele A, and then basically we say Fisher test and under my table, and it gets the p-value. This is just a very simple example using R. This is the Fisher exact test definition. It says that for group A, basically A, B, C, D are the allelic counts. And then once you have the A and B and C and D for each allele, then you find the row totals and the column totals. And then once you have that, then you can use the Fisher's exact test. This is the algorithm for the Fisher's exact test. It's a binomial algorithm, and it's the factorial. And also Fisher's exact test has two versions, one-tailed and two-tailed. So if you are interested in that, there are different variations of this algorithm. And the one which is very reliable is called two-tailed, and which is what I'm using for my algorithm. But this is basically the general fundamental concept for the Fisher's exact test. Then the P is the probability value, which gives you the value between 0 and 1. And the values which are closer to 1, they are not very important, basically. We are not caring about those. The ones which are closing closer to the 0, they are more interesting. Because the ones which are closer to the 0, they are rare. We are finding the rare mutations, but the ones which are closer to 1, there are so many of them, and we are not really interested in looking at them. We are looking at the rare situations that, exactly. Next, I will talk about what kind of Mapreduce algorithms did we use to solve this problem. By the way, this algorithm, we just did not sit down and invent this algorithm. This was a requirement from a pharmaceutical company called Sanofi, and then basically we worked with them to find this algorithm and present it. The first thing we need to do is that eliminate the duplicate records. Because in the allelic frequency algorithm, if you have duplicate values, then they will basically your calculation will not be correct. You don't want to have a duplicate values. So that's the first thing. And the second Mapreduce, you are just using the key of basically the chromosome start allele, and then using the Fisher's exact test to find the allelic frequency, the p-value. And in the Mapreduce phase 3, you are using the top 100 to find out what are the top 100 closer to the 0. That's another Mapreduce algorithm. The first one, basically the mapper. Let me come over here. This is to eliminate the duplicate records, very simple, right? Your key is chromosome ID, start group. The group means it is either group A or group B. And allele 1, allele 2 reference. So you basically emit key and value. You just send them to the reducer. And then in the reducer, you only select the mutation class with the highest mutation class. So you ignore all of the records which they have smaller mutation classes. So you select only one record which has the highest mutation class. And in phase 2, the mapper is basically the key is the chromosome ID start and stop. And the value is the group, allele 1, and so on, all of the attributes. And then you just emit key and value. Based on that key, the chromosome ID start and stop. So your key for all of the patients, you are looking for a specific chromosome start and stop position. And in the reducer, if you look at your key, the values are basically list of group, allele 1, allele 2, reference, and mutation class. And in the reducer, you set up all of your alleles for the group A and group B, and then find the frequency of table A, and find frequency of table B, and then you find the contingency table for all of your alleles, for that specific chromosome start and stop. So you form your contingency tables for all of the alleles, and eventually call the Fisher's exact test on the contingency table. And once you have the p-value, you can emit the value and the entire record. The Mapreduce phase 3 is after you generated all of the p-values. What are the top 100, which are closer to the 0? If you have a SQL query, we can do that. Select the star from Allelic Frequency table, order by p-value, and limit 100. But this is a very common pattern in Mapreduce. You can find top 100 or bottom 100 with a simple Mapreduce algorithm. So how do you do that? So basically, each mapper will find top 100, right? Let's say I have hundreds of mappers. Each mapper will find 100, right? Top 100, and then they will send their top 100 to a single reducer. So even though we are using single reducer, it is not going to be too much pressure on that, because all of them will send at most 100 values. That's it. So if you have, let's say you had 2,000 mappers, right? 2,000 times 100, it will be only 200,000 records for the single reducer, which is not much pressure. And I have found another pattern which is called monoid. You can use if your mapper generates a monoid, it's an algebraic term, then you can have let me show you that one. That will be here. This is the top 100. Given P up to Pn, top 100 will be smallest values, which are S1 less than S100 and so on. And top 100 for the allelic frequency means finding the smallest p-values, which are closer to 0. Okay, the top 100 mapper, each mapper finds its local 100 top and then sends to basically to the reducer. We will use as many mappers as possible. And top 100 reducer, it does the same as the mapper does. Basically, the mapper and the reducers are identical. What does it mean, a monoid? If you have a monoid, then you can have combiner. If you don't have a monoid, then combiners will not work. For example, if you want to find the median of a set of numbers, you cannot use, because it is not a monoid, you cannot have a combiner. This is an interesting thing that you can look at it. If any algebraic structure is a monoid, then you can have a combiner. Otherwise, it will violate the semantics. And this is the Mapreduce for top 100 p-values. Basically, I'm using the sorted tree map. And you might ask that why you are using that data structure. Because I only need 100 values. I'm not going to keep more than 100 values. It's never going to grow more than 100. And when it is more than 100, I just evict the one which is not proper. And then the reducer, it's almost the same algorithm. And here. And finally, in order to emit all of the values, we basically iterate all of the tree map and then emit the ones which are top 100. And next, I will show you that running the allelic frequency analysis. I prepare a shell script. I have a shell script which has two groups, Group A and Group B. I selected 100 by 100, basically, 100 in Group A and 100 in Group B. And then I run this program interactively. The name of the client program is the Allelic Frequency client. It just takes the zero, basically. What's that zero means? I'm putting the output at the file called zero, basically, somewhere. This is the example. So each bioset has ID. And that ID translates to a location of that bioset in the HDFS. So we have those biosets, and then once you have those biosets, then you can run this is the sample run. And finally, it runs and puts all of the p-values in a specific file that we designate. Thank you. And if you have any questions, I'll be glad to answer.
One question. Because you told that 95% DNA was same, and 5% only differ from one person to other person.
I'm talking about all human beings. Only 5% of our DNA are different. 95% are common patterns, and they are basically common among all of us. Only 5% are different.
So when you did this analysis, did you separate out those 5%?
That VCF file only focuses on that 5%. Good question. Yes. Yes.
For example, the data set that you mentioned, did I see that runs for 90 seconds?
This is just a very tiny sample. And the reason it took 90 seconds is because I have chopped up most of the data. I just wanted to make something run.
So what's a typical runtime for this application for a real data set?
As of now, I will not be able to talk about the numbers because this is a proprietary things for a customer. I do not want to bring the values on this here. So this was just basically fake data. Typical size of a cluster, we utilize several different clusters, and for the primitive, for example, the staging cluster, we utilize about 100 nodes, but for production nodes, we can utilize from, let's say, 300 to 600 nodes. Not in this case, no, we are not using, but there are several algorithms which unfortunately, I have not been able to find the Java version of those algorithms, and I'll give you one specific example. Cox regression algorithm. Cox was an English mathematician which developed this hypothesis. There's no package on the Cox regression algorithm, so I have no other choice than using the R in the, the reducers. For example, what I do is that generate all of the data and then call R once for all of the data, iterating, rather than I did this once, I generated for each reducer, I called R. The whole system crashed because R generates a process for every individual one. But if you collect all of your values and run once, let's say I have 1 million records I want to use R, right? Instead of running 1 million process for each individual one, let's say I create 10,000, 100 times 10,000, right? Then I put 100 files, each of them 10,000 records, right? Then that will be very fast. R will open that text file and crunch it very fast. So some of the algorithms we use R definitely because there is no Java equivalent of in the market. Those do not work well, trust me. We have experimented. Yeah, we don't use that one, but we just use the R directly. Pardon? Yeah, the only way we have been able to basically make this work is just partitioning data into segments and eventually calling R directly. But I think we need to look at maybe other big R packages and other packages too. I think we should, definitely. But if you try to run R as a separate for each individual reducer, it will definitely not work. Yeah. But R is a very powerful language, and also it's a golden, basically golden criteria for any bioinformatics people that they use those algorithms. And the accuracy of those algorithms, because it is implemented in the C programming language, the accuracy is much higher than Java programming language. For example, in some of the Java packages can create the accuracy up to seven digits, but the R was able to create accuracy up to 15, 16 digits. I'm not proponent of the R programming language, but it has lots of good things in it, and we need to learn and, to live that language with other languages as well. So I have only 1 minute. If you have other questions, I'll be glad to answer.
Is it necessary to do Fisher tests for every time when you it's basically test whether the means or whether the distributions are the same? So is it necessary to do it every time you compare the distributions of alleles in samples? Or are there some cases when you can simply say, yes, I have so much sample to the distribution, so I can say it's same without testing, because there are means and because the rows are so close that there's no need to test.
Yeah, but that's a very good question. But we are not doing any caching because these tests are run by different scientists and different physicians. So are you asking about caching the data?
No, about whether it is necessary to do the Fisher test each time you are comparing different.
Of course, I mean, you have to do that, right? Because the bioset selected at the different group, when you are doing the reducer, they will generate different sets of data. You have to. Otherwise, the semantics and the meaning will be lost. Thank you. Thank you so much.